latest edition of Scientific Discourse Friday Rooms. This is Scientific Discourse is a house that John and I put together to basically just talk about scientific issues of all shapes and sizes, including the philosophy of science and history of science. Talk about those, and the intent was that in this turbulent clubhouse atmosphere of a lot of people yelling at each other, we have created a safe haven of factual and logical conversations. So I hope that for those of you who are not members of the house, you would join it. And we have regular houses Fridays at this time, whatever time zone you're at. I'm Central Time, so 11:30 my time. John is Pacific Time, so 9:30 a.m. his time. This is a regular standing time, but outside of this, we also have rooms. And the rooms are hosted by either me, John, or Ben Tristam. John is a physicist, he's a retired professor of physics. I'm a PhD chemist who became a patent attorney and has been working in the pharma industry for the past 30 years. And Ben Tristam is an all-around technical guy. He knows everything about everything: electronics, physics, AI, computers, and much more. And Shamash also graces us every once in a while with his conversations on the philosophy of science and mathematics. We also have other guest speakers who come here from time to time and present. So this is basically a weekly what we call fun rooms where we discuss science. I run 3 different kinds of rooms. Chemistry rooms, where I talk about chemistry topics that are of interest. Pharma rooms, issues and science of the pharmaceutical industry. And then IP law rooms that basically mainly about patent law and other issues. And those are all on these Fridays, or if I feel like it, in the middle of the week at some point. As usual, there's no hand raising needed. Just unmute and come up on the stage, but please be thoughtful and respectful when you're on the stage. Anything, John and Shamash, you want to talk about before we start?
Just saying hi to everybody, and I'm following you. So whenever you move on the slide will move with you, so
Very
that you
good.
can present from your own. But you have to be in the document, okay?
Yep. Thank you.
I just joined Clubhouse, actually. This is my first meeting. I'm a physician scientist.
Well, welcome, Christopher.
Very nice.
Glad to have you here, and please join the Scientific Discourse Club and put this time on your calendar so we can enjoy your presence. All right, so we're gonna start with the talk. If there, oh, John, did you want to add anything?
Well, I'll only maybe use the opportunity to say I'm really glad to see Andy down in the audience. I don't know if he'll join us on stage. I haven't seen him for a while.
Yeah, I sent
Asking,
him an invite, and he also can
yeah, of
unmute
course.
and
Yeah.
come up. So talk if there. Oh, John, he's probably riding his bike through the tunnel, and so Anyway.
Yeah, and I'm in the car as I often am, so it may be a little bit sketchy too. Anyway, other than that, I'll shut up. Thanks, Sam.
So could you kindly give me a brief overview of what is this for?
Oh, so we discuss various scientific or science adjacent topics in this house, and the standing rooms are Fridays at this time, and either myself talking about chemistry or pharma or IP law. John talks about physics. Ben talks about AI, electrical engineering, etc. Andy, who just came up, is a mathematician, and Shamash is a philosopher of science. And so we basically put up rooms
And mathematician.
and mathematician. Sorry, yes. Anyway. All right, so today in April, in the last room that I talked about, we talked about the drug approval process at the FDA for a branded drug. If you go to the page of the house, you're gonna see the recording of that room in case you're interested. Today, I'm going to kind of extend that talk into the generic drug market and talk about how it is that that we get generic drugs. And so let's come here. And so on the second slide, which is really the first slide after uh, the title slide, I'm going to first talk about some vocabulary terms so we know what we're talking about. It's going to be a lot easier for the rest of the talk. So API, that's a very, very common word that you hear in the pharma industry. API is the active pharmaceutical ingredient. So it is the compound that exerts the therapeutic effect. So in a Tylenol tablet, the API is acetaminophen because that is the active pharmaceutical ingredient. A formulation is a combination of API with inactive ingredients. So those inactive ingredients are called inactive, not because they exert absolutely no function in the body, they exert no therapeutic effect on the illness or condition, on the targeted condition. So because of that, we say they are inactive. So for example, we have a lot of sweeteners, fillers, pH adjusters, things like that, that goes into a tablet in order to make the making of the tablet a lot easier, to make the ingestion of the tablet easier, to control where the drug gets released in the gut. Is it the stomach, the intestines, the colon, uh, and also they make the drug taste better, the bitter, so we don't have to swallow the bitter medicine. So sweeteners definitely have an effect on the body, right? Starch has an effect on the body, etc., but they're inactive because they don't do anything to treat your headache. Formulations come in different forms: the injectables, orals, drug A's, suppositories, and they come in. Drug A is a type of formulation that you inject the bulbous of the drug. So it's a drug in a polymer cage that you injected under the skin, and it just stays there for several months and slowly leaches out the drug. And after the drug is leached out, the polymer slowly degrades and goes away. So that's it has become a more common form administration of drugs, especially things that have to be administered for a long term. So basically, when you see a Tylenol gel cap versus a Tylenol capsule versus a Tylenol tablet, those are different formulations of Tylenol, and they're differently formulated because of their differences in absorbability and things like that.
So formulation is the final product, right?
Yes.
Everything that I ingest when I say I take my medication, we call it formulation.
Yes. So we say the drug is formulated as a tablet. Its formulation is the tablet, right? Yeah. The thing that comes out of the bottle and you put in your mouth is the formulated drug. The formulation is all the components of that formulated drug. Then three terms: branded drug, legacy drug, and listed drug. These all refer to the branded drug. So Tylenol is the branded drug, and it is a drug that is covered by at least one patent by the innovator. Innovator is the person who first introduces the drug into the market. So if the drug is being marketed under the patent of the innovator, then it is a branded drug or a legacy drug. Once the patent expires, then branded drug is being marketed under the drug's trademark. There's another term that's also used to refer to the branded drug, and that's ethical drug. That's a misuse of the term in this setting. An ethical drug is a drug that is dispensed by prescription, and as opposed to being over the counter. And it's called that because of the around the turn of the century, there was an ethics in pharmaceuticals or whatever to prevent snake oil salespeople from kind of populating the market, and so an ethical drug under that law was a drug that had a prescription, and therefore it was being ethically uh, dispensed. Today, a lot of people use the ethical drug to refer to the branded drug, but as I said, that's not entirely correct.
Sam.
So yes,
So, in the case of Tylenol, acetaminophen, is the branded drug Tylenol or is it
yes.
acetaminophen?
No, Tylenol is the branded drug.
And
Acetaminophen is the generic drug.
okay, and they get, so it's branded because the people that make Tylenol were the first to use acetaminophen.
Yes.
Yeah. Okay.
Well,
And it's
uh,
the same as
well,
paracetamol, right?
yes. So paracetamol is the European generic name for acetaminophen. Acetaminophen is the American one. And it has historical reasons. So basically, acetaminophen itself was never patented because it was discovered in the 1880s, and back then there was really no patents and pharmaceuticals to speak of. So what Tylenol did was it was the first uh, patented formulation of acetaminophen in a formulation that was very safe and properly measured and all that. And so they marketed acetaminophen under the brand name of Tylenol for quite some time, and after that it was under the trademark for Tylenol. So now when you go to the pharmacy part of the supermarket, all the generic ones just say acetaminophen, but the other ones say Tylenol. So what is a generic drug? A generic drug has the exact same API as the branded drug, but is not covered by any innovator patent. That is really the definition of a generic drug. Continue with this vocabulary thing. How drugs are named. We have several different types of names that a drug gets. So one is the brand name. The brand name is completely made up by the pharma company. And it is done based on marketing and this and this and that. And so, for example, the drug Lipitor. Lipitor doesn't mean anything. It's a lipid and just tor at the end of it. And so it's a made up brand name. Then there's a generic name for the exact same drug. And this generic name is also made up by the legacy company. So Pfizer, who came up with the name Lipitor, also came up with the name atorvastatin. And so the generic name, the only regulation really that it has is that it has to include the name of the class of drugs in it. So, or be somehow related to the structure of the compound. So here, the generic class is statins. And so all the generic names have statin at the end of them. So atorvastatin and simvastatin and things like that. So the atorva part of it is completely made up by the legacy company. And the generic name of the class is made up by the first pharma company who introduces that class into the market. So all of these names are made up names.
So class, yeah, you're probably going to get into this, but what, so the statin obviously refers to some part of the chemical compound,
Yes.
but it's something that's clearly kind of defines the kind of the standard function of all compounds.
No. Statin doesn't relate to the function at all. Is it me in the Matrix or John? I think it's probably John because it's driving.
John.
But okay, so statin doesn't signify the function. Statin signifies the core structure of the drug. So all statins have the same core structures. Some of these statins have cholesterol lowering abilities. There are a ton of statins that have no biological function, so they're not marketed or you don't hear about them. And so statin is a structure name, not a function name.
Are you hearing me now?
Yes.
Yeah, so quick question while I can. Sometimes it seems to me like you could have some drugs that share kind of a couple of already named structures. Does that just never happen? like a
Like what?
statin combined with something. I don't know. I mean, I just know so little that I have no idea. But I can imagine, like sometimes say, "Oh, okay, are we going to call this a statin, or are we going to call it a something else?" I don't know
Yeah,
what
yeah.
another class would be.
Yes, you run into that, and then the question is, how is it functioning then, right? Which core structure in there is the active core structure?
Yeah, so it gets murky.
Yes, it does. And so the FDA pays great attention to these names. So when I mean we talked about this that last time about the new drug application that the legacy company files with the FDA to get approval. In that application, there's also the proposed names for both the generic name and the brand name. And one of the first things that the FDA looks at is if you are handwriting this drug, can it be confused with another drug? That's
Wow.
the
They
most
take that into consideration.
that is the most important thing. The first
Wow.
thing the FDA considers is if the handwritten drug, if you write it in script, is it going to confuse the pharmacist as to what drug this is?
How,
So
how do they measure it? Do you have an idea? Like, do they get doctors and random pharmacists and then they tell them to write and read?
they themselves write it. Yeah. And you know, they're, yeah. And, and, and once they say this is a confusingly similar script name, you're SOL, right? You can't appeal it because it is the mistakes in dispensing medications are common and fatal, and so the FDA does everything in its power to stop that. And
Surely
so,
we must be on the verge of the FDA saying no handwritten prescription
well, yes,
at this point.
we are, but then the FDA also worries about the verbal. If the physician is saying to, you know, the PA, put this in,
Oh yeah.
uh, will the PA then confuse what the physician said? So these are you know the things that the FDA is the number one thing with respect to the name that the FDA worries about. After that, it goes and figures out, okay, is this really the right class, etc. But the third name is the IUPAC name. IUPAC is the International Union of Pure and Applied Chemistry, comes up with all the rules of chemical symbols, names, nomenclature, things like that. And based on that, you have that long chemical name for atorvastatin. And so that name is basically equivalent to the structure. If I know that name, I can draw the structure. And if I know the structure, I can write that name if I know the IUPAC rules. So in the package insert, you will see all three of these names spelled out in different parts of the packages. All right, so now that we have the vocabulary out of the way, let's talk about generic drugs. And 1984 is the critical year in the generic drug industry. What happened before 1984? And I should say, really, before 1984, we didn't have a generic drug industry to speak of. Uh, the laws, both the patent laws and the FDA laws, basically prevented generic drugs to come into the market. On the FDA side, there was data exclusivity, which means that another drug company who wants to introduce a drug with the same API as a previous drug company couldn't rely on the data of the previous drug company for their approval. So basically, this is the rule today, is that I can say if I'm coming up with generic atorvastatin, I can just say atorvastatin is safe because Pfizer went through safety analyses to the point that got FDA approval. So I can rely on that and say FDA at this point decided that atorvastatin is safe. So atorvastatin is safe. I don't have to prove that anymore. Whereas before 1984, you couldn't do that. Data exclusivity prevented other companies from relying on the legacy drug companies' data. The other thing was patent loss. If you were to do any experiments on the API while the patent was pending, you would end up infringing the patent. So companies, generics, had to do complete clinical studies after the patent on the drug had expired. So complete clinical studies take between 5 to 10 years. So this effectively extended the patent term on the drug for those 5 to 10 years because the patent had expired, but nobody could enter the market. And so the end result was that in the US, only 10 to 13% of drugs that were off patent had a generic equivalent in the market. And those 10 to 13% were drugs that were expensive enough that would make sense for a generic drug company to conduct full-scale clinical trials and enter the market. This, of course, had amazing consequences. One was that the non-US drug companies enjoyed a global windfall, right? So who's distributing drugs in Africa? Who's distributing drugs in South Asia? Who's distributing drugs in South America? It's not going to be the legacy drug company because their drugs are too expensive for those markets. So who's going to do that? A drug company in India, a drug company in Israel, a drug company in Egypt. Why is that? Because those scientists are all US trained. And then they go back to their countries and they're like, hell, there are no drug patents here, right? So we're going to set up a lab. We know how to set up a lab. We know how to synthesize things. We know how to formulate things. We learned all of those from the US. And so we're going to come here and make those drugs and sell them in Africa where there are no patents. Sell them in South America where there are no patents. Oh, there are patents? You're going to sue us there? Good luck. And so the US companies were completely shut out of the generic drug market because they would now be infringing in the US, a US patent of a US drug company, and those lawsuits are going to be horrendous. And so the US drug companies were shut out of the generic market in the US and all the drug distribution markets globally. And this was bad for business. And if it's bad for business, it's bad for America. US consumers were unhappy as well, right? because they didn't have access to cheap generics. They look across the pond and say, the same drug that I'm paying $50 for in Namibia is being sold for 50 cents. Why is that? Why can't I have that 50-cent drug? And the reason is, oh, it's 50 cents because the Indian generic company manufactures. So into this comes the Hatch-Waxman Act of 1984. It completely upended the pharma industry in the United States and by extension in the world. It really started the generic drug industry in the United States, to the point that today the majority of drugs sold in the United States are generics, whereas we saw before Hatch-Waxman Act, it was close to 10%. And who are Hatch and Waxman? The two most unlikely partners in Congress. Orrin Hatch, famous senator from Utah, Republican, was in the Senate for 40 years. Mitt Romney famously replaced him. And Henry Waxman, he was a Democrat House member from California, ran the Governmental Affairs Committee in the House for a long time, and he was kind of famous for going after Republican administrations breaking the law. But in 1984, these two guys, who were both on the Judiciary Committee, came together and wrote this piece of legislation. And I should kind of preface this by saying that this is my absolute favorite legislation in the annals of the development of the US Code. I don't think any other piece of legislation was as impactful and as successful as the Hatch-Waxman Act was. And we'll talk about this. The law had these 5 stakeholders: the legacy drug companies, generic drug companies, insurance companies, the federal government, and the consuming public. And all of these stakeholders benefited greatly as the result of Hatch-Waxman, and those benefits were intended benefits of the law. So it was one of those laws that did exactly what it said it would do and had exactly the same results that it said it was going to have. So what does Hatch-Waxman do? For the legacy drug companies, it shortened the data exclusivity time to 5 years, but said that within those 5 years, the FDA will not entertain any generic drug company or any other branded drug that has that API in it. So for 5 years, no one else can get approval on that API. And so I say 5 years. 5 years is for most drugs. If this is an orphan indication means that there's less than 100,000 people in the United States who are suffering from that disease. For an orphan indication, the data exclusivity period is 7 years. For repurposed drugs, meaning that the drug has already been approved for one indication, you are introducing it for another indication. Let's say aspirin was in the market for headache. you're introducing it to prevent heart attacks. That's a different indication. For that, you get 3 years of exclusivity, but 5 years is basically the typical part. That gave drug companies a great deal of ease of mind because they knew that for those 5 years, nobody's going to come in. Then they also extended the term of the patent on at least on at most one patent that covers the drug, and basically you get back a portion of the time that the drug was in the FDA going through the approval process. The time that you get back has a very complicated formula. I didn't think it was necessary to go through that, but just know that you get some part of it back, and so we play at the patent office because patent office also has patent term adjustment term, meaning that if the application gets delayed in the patent office, then the length of that delay is added to the term of the patent, and so we play games to force the patent office to make a delay, and then you know, and then add the patent term restoration to that, so you get a much greater patent term. And
Sam.
then, yeah,
Um, oh,
go
so
ahead.
you don't say explicitly here what happened internationally. Did this change anything about the generic market internationally?
It basically knocked off a bunch of international generic drug manufacturers off of their pedestal. They were replaced by US companies.
Just because of the shortening of the patent term, or what? I'm not sure exactly. I mean, you still have 5 years
So
when companies can
let's
do this, right?
no, let's
No.
it's not all this. The main part is that data exclusivity and the 5-year data exclusivity and what happens after that expires.
So the
And
point is that nobody knew what the formulation was for 5 years.
they knew what it was, they just couldn't say that, well, this other company proved this to be safe, so we're going to take the fact that it is safe and introduce this other one. The FDA was saying that
Oh
no, you have
no, but
to,
I
you
mean,
yourself have to prove it's safe
a
also.
company in Egypt can market to Africa, right?
But they couldn't do it in the US.
Not right, but I mean, they still have that huge market
Yes,
in Africa that would otherwise, if somebody in Africa wanted it, they'd have to get the U.S. drug, right?
but India was giving them the US drug. Israel was giving them the US
Hmm.
drug.
So nothing
In
changed as far as that goes.
sorry, before I'm a little bit confused
Well, I guess
as to the
I'm just
question.
thinking. I mean, it just strikes me that a big part of the complaint before was losing U.S. drug manufacturers losing international markets, and they still do, right? I mean,
No.
No.
See, that was the thing. I mean, they were losing the international markets because they couldn't manufacture the drugs here in the US and then take them and sell them. So they would have had to set up labs, manufacturing facilities outside of the United States to do this. And that would have been extremely expensive, so that it was cost prohibitive.
Oh, I see. I
And
guess I thought that was already happening. I thought the drugs were
no,
being manufactured outside the
not
U.S., sold outside the
not by the
U.S.
US companies. They were being,
But the U.S. company loses that market because some other
yes,
company does it.
yeah.
Yeah.
So that was the reason they wanted the law to change, so they could basically do all the testing and manufacturing here in the U.S. and then ship it uh, international. I don't know if I addressed your question or not,
Well, I'm
but
still not quite sure I understand. It seems to me that you saw the
so
case. So the
let's
drug is introduced, and a company in India says, "Oh, we're going to make that drug and sell it," and they do.
yeah, basically.
And that's bad for the U.S. drug manufacturer, who then doesn't get to sell it to the people that the Indian company is selling it to.
Yeah, but so there are other things also, right? So at around the same time that Hatch-Waxman in the United States is kind of getting very strong, we see that India in the early 2000s changes its patent laws. Up until then, pharmaceuticals could not be patented in India on ethical reasons, and this is why India had some of the largest generic drug manufacturers, because you could not have a patent on a pharmaceutical. Well, in the early 2000s, India changed the law, right? So now U.S. drug companies always file in India, and so that has kind of slowed down their entry into the market, which has allowed the US companies to fill that gap. Then the other thing that has happened is that legacy companies in the United States have set up generic arms. So when the patent runs out, we're going to get into exclusivity period for generics now. They're going to be the first ones. The same company, right? Pfizer will now introduce the generic of their own company, of their own drug. And
Mm.
so they get the exclusivity for that. So there are, it's not just one thing that changed. So globally, a lot of things changed too.
It's really fascinating. I find. Good morning, guys. Good morning, everybody.
Good
Good morning,
morning.
everybody. Just throwing a general thing, just what came to mind. It's funny when I was young, was like in my 20s in Tokyo, and I didn't have much money. I would like sign up for like, hanging out at the hospital, and they needed Hakujin-san, right? They needed white people to do some tests on. So I would make like, $2,000 just hanging out in a hospital. Okay, pump me up with whatever you want, and see if the drugs work. It was kind of a fun time, but boring as heck. But yeah, that's always interesting. What are the trial tests? But it's really fascinating how different nations really compete against one another, and how they demonize one another to like, the whole big thing now is in Canada we have the electric cars coming in from China, and
All right,
y-
let's keep it to the generic drugs
oh,
if we
to
can,
the drugs
not
issue. Okay, I'm
the,
sorry,
yeah, not
yeah, but
yeah,
it's
because
interesting how
we
the
have a
nations
lot to
compete against each
cover,
other. Yeah,
so
sorry.
I don't want to like
I apologize.
go
I
on
apologize.
tangents. Yeah. All right, so the other very important thing that the Hatch-Waxman did was the introduction of the Orange Book. We'll go through that in a little more detail, but it's basically the company gets to list patents that cover the drug. And so what this does is that it hugely increases the barrier for entry for generic companies, and we will see shortly why that is. So patent laws, section 271A defines what an infringement is, which basically means whoever makes, uses, offers to sell, sells, or imports into the United States without authority a patented invention, then they infringe the patent. So does this apply to generic companies who are going to do their clinical testing on the branded drug? Before 1984, the answer was yes. But as part of the Hatch-Waxman Act, a 271E1 was added to that, which is the safe harbor provision. And it says it shall not be an act of infringement to make, use, offer to sell, or import into the United States, right? The same things that for every other invention is an infringement. In this case, it's not an act of infringement if the infringing activity is done solely for uses reasonably related to the development and submission of information under a federal law which regulates the manufacture, use, or sale of drugs or veterinary biological products. What does that mean?
What?
So yeah, I kicked them out. Yeah, they're assholes in the world. So basically what it says is that if you are using a patented invention for the purposes of generating data to submit to the FDA, then you're not infringing. But if you do the exact same thing for any other reason, then you're infringing.
So you can use it for a clinical trial or something like that?
Yes, absolutely. Or for formulation development, for clinical trials, for anything that you want. And this has gone up to the Supreme Court a few times. And every time the Supreme Court has broadened the interpretation of reasonable relation. So basically, if you're doing an experiment just to determine that such and such drug doesn't work, it is not. It's a comparison with the drug that you're trying to develop. The Supreme Court has said that is reasonably related to, because at the end of the day, you're going to use that information to generate the data that you're going to submit to the FDA. So that's a very, very, very broad interpretation. It doesn't apply to uh, uh, assays and things like that. Those, but in terms of using the compound, it's pretty broad.
Does it apply to work product too?
What does that mean?
Like the process and everything that comes along while you're in the process of doing a clinical trial
Is
or
it
something like that? Any
if
product that comes out of it? Like not
if
necessarily just the goal product, but any incidental like discoveries along the way?
if you are using a patented method in order to test a compound where the results will be submitted to the FDA, then that's not the infringement of that method patent. So, all right, so what is the effect of the safe harbor? It just means that generic manufacturers can conduct clinical studies and manufacture their drugs while the patent is still pending. They can't sell it, but they can't distribute it to the pharmacies, but they can do everything else. And so we routinely, I mean, this is a normal thing, that if the patent expires on a Tuesday at 12:01 a.m. on Wednesday, what do you guys think you're doing? Huh? Do you think like
Drugs.
you're smart? Do you think you're funny? What
Drugs.
the fuck? I mean, why don't you have any respect for
Could you
yourself?
turn hand raising on because we're streaming on YouTube?
Oh, okay.
The
Yeah.
other guy. the other guy started playing copyrighted music a minute ago, we could get banned for that.
All right. I'm sorry, but I guess I can't trust people to be mature. Anyway,
You knew that, Sam.
so basically, I mean, this is a very, very common scene that at 12:01, trucks leave the distribution center to go drop off the drugs at pharmacies. So before this, the generic company has made all of its contracts with distribution uh, paths, distribution routes. They have all their agreements with pharmacies. They have all of their agreements with insurance companies. All of that is set before the patent expires, and so patent term is not now artificially extended. So the day the patent expires, the next day you get the generics in the market.
So, Sam, you were saying earlier that the legacy company will in the after that will typically market its own generic product too, and so it really is the case that at that point they're just hoping some people will really want the original branded name so much that they'll pay an extra dollar or two or three or whatever it is. I mean, that's really all that's going on there, right?
Well, at this point, it doesn't really matter what the patient wants. It matters what the insurance company pays for. And so
For a
once the
prescription drug, right?
yeah,
Yeah.
so once the generic hits the market, then you actually have to make a case for the branded drug.
So, but let's
If
just take the case of Tylenol. Does no, who is that? Pfizer? Who makes Tylenol?
I think it's P&G, Procter and
Well,
Gamble.
whoever it is,
Yeah.
do they have? Do they also market a generic drug that is not
Oh yes.
doesn't have the name Tylenol? And so, but
So when
people,
you
and it's
go to
exactly
like
the same,
Safeway,
right?
when you go to Safeway and you have the Safeway brand of Tylenol, that is almost always made by the brand name
Oh,
manufacturer.
really? Oh,
Yeah,
I see. I didn't know that.
yeah,
Okay, it is
yeah. But
exactly the same.
uh, no,
How so?
because they don't take as much care in the formulation. So even though the API is the same, the absorbability may not be the same. And this is one way that, the brand name company introduces a subpar generic so that people use that and say, oh, it didn't work. And then you go and use the branded uh,
And
drug.
there could be good reasons that it wouldn't work as well, just because of the observability, rather than because even though it has the same API.
Well, the thing is that it's your formulation that has the biggest factor in whether the drug gets absorbed or not.
Mhm. It's
And so if they don't do everything that should be done to give you maximum absorbability, right? Then that's fine. But note that Tylenol is a bit different because it it is not it's an over-the-counter drug. It's not an ethical drug. So the FDA has kind of less uh, uh, kind of supervision over that than it does over prescription drugs. So on slide 13 now, uh, how does a generic manufacturer go before the FDA? We saw that for a legacy company, they first filed an investigational new drug application in order to be able to start the human clinical trials. And then at the end of the clinical trials, they file the NDA, the new drug application, not the non-disclosure agreement, but the new drug application. And that's the official application to get approval. And once the NDA is approved, the drug hits the market. Generic companies file an ANDA, which is an abbreviated new drug application. And it's abbreviated because it relies on the data that's in the NDA. So this is filed after the data exclusivity period has ended. So now a non-legacy company can rely on the data on the NDA for their clinical trials. So you can file your ANDA anytime, but the FDA won't look at it until the 5-year data exclusivity term has terminated. So what goes into ANDA? ANDA has to make certain certifications, and most of them are technical. A couple of them are legal. And the certifications are as follows that the manufacturing process assures and preserves identity, strength, quality, and purity of the listed drug. So basically, they have to show that in their manufacturing process, the compound that comes out at the end has the same identity, strength, purity as that of the listed drug. So in the listed drug, we said is the same as the legacy drug. In the legislation, it calls it the listed drug because it starts out by saying that a drug that is listed under such and such provision of the law, and that means the branded drug. And so when it says listed drug, it's kind of a drug that's listed on the list of branded drugs, basically. Conditions of use are the same. The API is the same. The route of administration and dosage form, strength, everything is the same. And the drug is bioequivalent to the listed drug. Okay. So note here that formulation doesn't have to be the same. It can be formulated differently, but has to be the same dosage form. Meaning what? Meaning that if the legacy drug is a tablet, the generic drug also has to be a tablet. If it's an injectable, this also has to be an injectable. So the dosage form is the same, but what goes into the dosage form, meaning the formulation of the dosage form, that can be different if it gives bioequivalence. What does bioequivalence means? It means that after you administer the generic drug at the same dose, the same route of administration, the concentration profile of the generic drug in the blood is exactly the same as the concentration profile of the legacy drug,
So
meaning that, go ahead.
does that mean that, for instance, time release formulation would have a different would not be
Yes.
bioequivalent? Yeah. Okay.
Yeah. So you if you want to go time release, there has to be a legacy time release so you can have the generic of that. So the three main uh, numbers that they look at, one is Tmax, the time after administration where you get the maximum concentration in the blood. Cmax, the maximum concentration in the blood, and AUC, which is area under the curve, the total amount of drug that was absorbed. So those are the three main ones. There are other factors that pharmacological factors that you look at for bioequivalency, but if you hit those three, you're 99% of the way there. What about safety and efficacy? We talked about that. You rely on those. You don't actually get to see the data, but you say, because my drug is the same and is bioequivalent, therefore necessarily it has the same safety and efficacy profile. All right. This is where rubber meets the road. Up until now, there is no reason for anybody to sue anybody. Now we're gonna get where the generic drug lawsuits take place. You have to, the ANDA filer, the generic drug company, has to make one of 4 certifications with respect to patents. The first one is that no patent information is listed in the Orange Book. We said Orange Book is the name of the database where the branded companies list all the patents that cover that drug. So you say there are no patents listed in the Orange Book for this drug. This is not controversial. It's objectively ascertainable, so there's no lawsuit there. Or you can certify under paragraph 2 that the patents are expired. This is again objectively ascertainable, no lawsuit. The generic company, paragraph 3 is generic company plans on marketing the drug after the patents have expired. So the branded company just waits to see if the generic drug did exactly that. And if not, then you have a simple case of infringement. Paragraph 4 certification, that is the meat of this whole thing. This is where millions of dollars are spent in litigation. This is where companies try and stop the generics from coming in. So paragraph 4 certification says the patents listed in the Orange Book and not expired are not infringed or are not valid. And so this is where people like me come into the game. I have written countless of these paragraph 4 certifications, is that you do a complete legal patentability analysis or a complete non-infringement analysis on the patent. And at the end, you say that, based on the foregoing, you are not infringing a valid patent. And with that, the ANDA filer certifies the application under paragraph 4. So slide 17, we see that, and so remember that safe harbor one, that you could do everything that you want and you're not going to be infringing. Well, this is where things change. And ANDA filed with a paragraph 4 certification is an act of patent infringement. So once you file your ANDA, you are out of the 271E1 protection, the safe harbor provision protection, and now you are liable for patent infringement. So what happens is that you receive the paragraph 4 certification from the ANDA applicant. They're supposed to provide you notice, I don't know, within a couple of days of filing the ANDA. And you say their paragraph 4 certification is hogwash, and you have 45 days to file an infringement suit. While you file that suit, the FDA stays the consideration of the ANDA application for 30 months, 2.5 years. So as soon as the legacy company, so let's say I was involved with the Lipitor litigation, one of the worst mistakes of my life. We represented Ranbaxy, the Indian generic drug manufacturer, against Pfizer, and so the day ANDA was filed, that day Pfizer sued, and so for 2.5 years, Ranbaxy couldn't get into the market. So this is one of the other things, one of the other important tools that the legacy company has to keep the generics out of the market. So all of this is from the Hatch-Waxman thing. One more thing I think I forgot to put in here, and I'm sorry, so I'm going to say it verbally, is that the first generic company who files an ANDA and their ANDA gets approved gets 180-day exclusivity. So that the first generic company who files an ANDA gets 6 months exclusivity in the generic market. And so this is why some of the legacy companies have their own generic manufacturers. Why? Because they have the ANDA ready to go, right? They have all the data, they have everything. It's just they're going to change the name to a generic name. They file it, and now they added 6 more months to their patent term. Because they know when things are going to happen exactly, and so they can be the first to file the ANDA application.
Sam, when
Yeah,
the
any
certi-
questions so far? Yeah, I was going to say.
so the back of the certifications, um, uh, it made it sound like all four of those paragraphs had to be there, but
No,
four doesn't have to be there if there is simply nothing.
those are ORs. The certifications are ORs, not ANDs.
Okay,
Yeah,
okay,
you
yeah,
certify
okay, okay,
under only one of those paragraphs. Yeah, you
okay, that
can't do
makes
under all four. Yeah, yeah, it's only under. So, any questions before I kind of switch a little bit and talk about the Orange Book?
It seems weird a little bit. I guess it happened in the case of Lipitor, but it seems a little weird that it wouldn't always be the legacy company filing the ANDA, and so those paragraph four things would basically never happen. Or I guess you can have a paragraph four at the same time that you have a different one or something. Maybe I'm not. I need Shamash to help me with the logic here or something.
No, nobody can help you with the logic because we're not privy to their internal business considerations, right? They may have thought that they don't want to go that they don't want to give resources for a generic atorvastatin and take those resources away from development of newer drugs. There are a lot of these considerations that happen. We don't know about. We don't know about agreement. Behind the scenes agreements between the generic company and the branded company. Those agreements can't be very obvious because that's an antitrust violation, but do those collusions happen all the time? They're careful about how they're colluding And so you can't just based on the visible facts decide whether the action was logical or not.
Hey, also, I'll just mention Austin has a couple of questions in the chat. I don't know if you want to take time to read them now, but maybe at some point. I don't.
There are always class action mass torts against generic companies. There are always these litigations against all drug companies. And sometimes they go forward, sometimes they don't. But as a rule of thumb, you have to kind of consider that drug companies that you have heard of are doing at least 10 or 20 litigation on any given day, uh, all year long. And so some of them are mass torts, some of them are not. So who do the large pharmacies hire to assess the absolute? The pharmacies don't hire anyone to determine those. They rely on the package insert that the drug company sends out with the drug, and the package insert is based on the data that's submitted to the FDA. So once the drug hits the market, its package insert is publicly available, and so pharmacists basically go off of that or based on the information that the pharmacy reps have given. All right. If there are no other questions, I'm going to go to slide 18. What is the Orange Book? Well, Orange Book, the real name is Approved Drug Products with Therapeutic Equivalence Evaluations. That's the name of the publication. Before the internet, this is like the mid 80s, right? It was published as a book whose cover was orange, and so it was called the Orange Book. It's published by CDER. CDER is a Center for Drug Evaluation and something else. It's one of the offices at the FDA. So in the URL, you see Access Data, FDA, Scripps, CDER, and then OB. OB's Orange Book. But everybody calls this the Orange Book. Nobody calls the approved drug products with therapeutic equivalence evaluation. So if you go to that URL, you see the search name for that, and you can put in where it says enter at least 3 characters, put the name of the drug. So I did this with a drug called Xifaxan. I searched for rifaximin. Rifaximin is the generic name for Xifaxan. You can see here in the entries 2 and 3, Xifaxan is named as the proprietary name. And then if you click on one of those, you get to this page, which gives all the information about the drug. I mean, all the information you need to know with respect to the filing at the FDA. And underneath at the bottom says patent and exclusivity information. You click on that and you see these are all the patents that cover that drug form. What's the drug form? A tablet of rifaximin at 550 milligrams. It is covered by all of these patents. Under the exclusivity data, it says there's no exclusivity data because it is past 5 years from the filing of the NDA. So the exclusivity period, the FDA exclusivity period has expired, but the patent ones are not. So you want to know if you can certify against this, you go ahead and you download one of those patents. This is 8193196. And you say, holy shit, the attorney agent is Sam Tahmassebi. And yeah, that's why I had this example. So with this, this was, if I go back to slide 21, it tells me that this patent is going to expire on September 2nd, 2027. And so if I need to make any certifications under paragraph 3 with respect to this patent, I have to say that we're not going to ship the product before the 2nd of September of 2027. So that's the Orange Book. The last two slides are basically, so what happened with the Hatch-Waxman, starting with slide 23. So generic drugs are cheaper than the branded drugs. By how much? By at least 80 to 85%. That's a huge drop in prices. And they're more prevalent. As soon as the generic drug hits the market, within 6 months, they had 80% of the market for that drug, and within 2 years, they have 97% of the market. So basically, the entry of the generic drug is the death of the branded drug. And so the bigger market share at lower prices leads to overall lower prices for the consumers. So 4 decades later, before 1984, we had Teva, Dr. Reddy, Ranbaxy, and some other companies as the largest generic manufacturers in the world. We didn't have any US companies in the top 25. Today, US companies occupy much of the top 10 slots, including 2, 3, and 4. Teva Pharmaceuticals in Israel still is the #1 generic manufacturer in the world, but we have knocked off Dr. Reddy and Ranbaxy from the top 25. The cost of drugs is still skyrocketing. We all know that. That's a huge problem. But the rate of increase is a lot less than it would have been without the Hatch-Waxman. So we see with Hatch-Waxman, prices have come down or are not increasing as fast as they would have been. So consumers win. We see the pressure on the FDA for review of these applications is a lot less because these applications have become abbreviated. There's reliance on old data. So these applications get approved a lot easier and a lot faster. So that's better for the FDA. We've seen generic drug companies can enter the market immediately, and it has been really good for the generic drug manufacturers. And the benefit really for legacy companies is that they have 5 years of peace of mind. And after that, if they can manage the life cycle of their drug with patents, which is really what people like me do, then they can use the tools of Hatch-Waxman to stave off generic entry for much longer than before. Another benefit of this for consumers is the pace of innovation. As I said before Hatch-Waxman, about 10 to 15% of off-patent drugs had a generic equivalent to it. So companies were not really worried about their drugs going off patent. They would not lose market share. But now that they are losing market share when it goes off patent, they are more incentivized to come up with better drugs, newer drugs, so they can continue their exclusivity in the treatment of the condition. And so the game that's being played is that, yeah, there's that generic equivalent, but the new branded drug works a lot better. So people still go to the branded drugs. Uh, and that has been much better for the patient population, that they're getting newer uh, newer medications at a much faster rate because the generics are breathing down their necks. Well, this concludes what I wanted to say about the generic drugs. Thank you very much for listening to me for the past hour and a half. And I'll be more than happy to answer any and all questions.
That was really, really interesting, Sam. As always, I'm always surprised. I kind of think, oh, is this going to be interesting to me? It's not something I pay any attention to, but I'm struck by all of the thought and the logic and, all of then the legal, just the legal precision and all of those things. But I'm also struck by the fact that you seem to be saying that, government intervention and regulation actually was a plus for the consumer when we all know that, in fact, regulation is a bad thing. You should just let the companies duke it out, right? We'd all be better served if it was a
I
purely competitive market.
I don't get into the government regulation is good or bad debate generally, but with respect to the FDA, I would say that the FDA has saved so many millions of lives by virtue of its regulations. I cannot be convinced that the FDA is a bad agency. Yeah, it doesn't work as efficiently as you want to. There are cracks, but there are cracks in everything, right? But when you look at it overall, the effect that the FDA has had, it has been tremendously positive. And it is extremely sad for me to watch the FDA being dismantled under the current administration, because
Yep.
there's a clear lack of understanding of what the FDA does. I mean, one of the things that kind of upsets me with the anti-pharma, anti-science is that when you look at these processes, right, the the least educated person in this whole process is the lawyer. Everybody else has PhD and MD PhD, right? And they've been working in this field their entire lives. And for someone who hasn't gone to college and can barely spell their name, come out and criticize the pharma industry is something that I cannot accept, because you cannot get any better than this globally. You cannot get any better than this. No other country in the world does drug development as well as we do. Oh,
Are you referring to the lawyer Robert F. Kennedy Jr.?
No. I was talking about the entire process of, you know, starting from we had a hit on a target in a cell until our NDA was approved, our ANDA was approved, now generics are here, right? In that entire process, in that 30-year process of all the people who are involved in making decisions and getting the drug forward, the least educated one in that group is the lawyer on the team, because the lawyer doesn't have a PhD generally, right? Now, I mean, nowadays it's a lot different. You get a lot more people like me as the lawyer for these companies who have both a PhD and a law degree and experience in industry. But generally, we're the least educated, the dumbest ones in the room. Those scientists run circles around us.
What
I'm
do you what do you think of lawyers like Robert F. Kennedy kind of running policy or putting his 2 cents in?
you know the question is not that he's a lawyer, because really all of our HHS secretaries have been kind of lawyers or politicians, but like Tommy Thompson or uh, you know, you get, I forget who the other ones were, but you get a lot of people who were governors or senators or something like that. Because those guys are not making they're not making science decisions, they're making public policy decisions. And you don't want a scientist to make public policy decisions. You want the scientist to provide the data for the politician who's going to make a public policy decision, because this is not all driven by science. There are a lot of other uh, things that you need to take into account, and that's the job of the politician. So I'm not per se upset that a lawyer is in charge of the HHS. I'm upset that a lawyer who doesn't listen to scientists is in charge of the HHS.
Sam, what implications are there for like the legal issues when it comes to using AI in development of medicines and pharmaceuticals and stuff like that?
I don't know. Uh, we're not there yet, and so I can't predict.
Do you think that that's going to be a big issue? Like, I mean, we're going to have to create some kind of standards and stuff like that. There's all kinds of things that are happening with like stem cell research and stuff. When we got to the point where we could do that in cloning, we had this big like throw on the brakes kind of moment because we didn't know what we were going to do or how we were going to police it or and all that stuff. There was this gray area. Do you think we're going to have that with AI as well?
Well, the thing is that you cannot get drug approval based on AI alone, right? At the end of the day, you have to have clinical trials and show how the drug performed in humans, and that is not something that AI can do. So I would say no. I mean, AI may go into the initial stages of drug development as it has been. It's not a new thing. I mean, we've had companies who used to use crystal structures of proteins and then use AI to design compounds that would fit into the active site of these proteins. So So those types of things have been going on, and my guess is that in data analysis, AI is going on. I'm in-house counsel at a company where we use AI to and machine learning to do proteomics on cells. That is something that humans couldn't do. Just like, okay, I'm changing these conditions. Tell me what correlations do you see in the proteomics? And that was something that humans could not do. So on the one hand, this gives drug developers a huge tool in finding out correlations that didn't exist before. On the other hand, you have to be cognizant of the fact that it might be hallucinating, that it's coming up with correlations that actually don't exist. So, but the thing is that if you're coming up with correlations that don't exist, you're going to fail in future steps.
Yeah, but like when it comes to like owning the product and stuff like that, like if they do, if you have companies that are collaborating with these AI giants to crunch data and to make models and stuff like that, is there going to be some kind of like royalty or ownership like scheme or structure that's going to be created for that on the legal side? And is it going to impact the cost of the drugs for people?
The answer to that question is really nobody knows, because we we can't predict what issues will come up during the use of this. I mean, I kind of look back in 1995 when Netscape had its IPO. It was at that time it was the largest IPO that there was, and people were saying all these things about how the web is going to destroy everything. And at the end, we saw that all the issues that people talked about in 1995, none of them came to pass. Instead, issues came up that nobody could predict in 1995 that someday this will be an issue. And I think AI is in that stage, is that we are not thinking in an AI dominated world. We are thinking in a non-AI world, and we're trying to kind of put that AI thing into this world. And it's like, no, that is going to be a completely new world. And so I really don't know what issues are going to come up and how those issues are going to get resolved. We just have to see, we just have to follow the litigations and see what the
It's
issues are going
it's
to
gonna
be.
it's gonna be a whole new mindset. You're right about that. I just one of my concerns for it is like it's specifically related to clinical trials and drug development. If they if they if they create a contract with people to with an AI company to crunch some of the numbers and to like run some models and things like that, I worry in some ways about the privacy of the people in relation to that. The privacy, that's one of my concerns. And the other concern I have is like these AI companies getting like uh, stock options and things like that in the companies or stakes in the medications that they're working on, and then turning around and utilizing their formats to maybe more promote those treatments. Like the ethical problems are really uh, clearly apparent to me when it comes to that. Like because AI reaches so far across the divides, and I think there are like issues related to that that we're going to have to encounter and have litigation over.
Yeah, I think these things are right to worry about, but I don't know if by worrying about them we can come up with any solutions, because, as I said, we don't know what the problem's going to be. So I have kind of a wait and see approach to this. It's kind of like, all right, once it gets to a point that affects my life, then I'll do something about it. Or if I can't do something about it, then I'll adapt. I mean, the thing is that AI is here to stay. You can't you can't say, I'm not going to do AI in drug development. If you say that, you go bankrupt immediately. It's here to stay. It's giving you an advantage. It's giving your competitors an advantage. So you have to use it, right? But then how do you use it? What do you do with information? How, what are the shortcuts that you're taking using AI? Those are all, those to me are the important questions rather than are you using AI? But how are you using AI? And what do you do with the result of it? For me, is a bigger question than whether you're using AI. But then again, Ben knows I don't understand AI, so maybe I'm completely off my rocket things.
Hi, I just come up. Thanks for letting me just go on the stage. I'm actually can help. I'm the CEO of an AI health company
Oh,
and also like a scientist more than 20 years. And
welcome.
our yes, and I'm happy to explain everything. I'm working obviously with AI, but we are established our program, a large language model, since 12 years. So long time people didn't know what AI is. So we're since 4 years certified and we're live on 7 hospitals right now. We're also fully reimbursable and we're saving everyday lives for sure. So AI had definitely changed the speed we're working on, things which took before 4 to 6 weeks from diagnostics and treatment selection. We break it down to 1 hour to a ridiculous low price. So health insurance companies are trying to really signing us. We're getting overrun with customers. So we even don't need any seed or any funding because we are complete bootstrapped us in because the demand is so high. But we really change a little bit everything around us. So, for example, a cancer patient goes before the doctor, they give blood, there will be a genomic analysis, and what we do then it sends to 30 bioinformatician, 25 genetic counselor, and it takes 4 to 6 weeks until they come up with the causal gene, so that gene which is responsible for the cancer or for the rare disease. And with AI, we can do this in literally 1 hour. And with 97% accuracy, a human could never achieve. And we certified it and tested it in 100,000 patients. So it's insane accurate. And now patients can actually get a treatment immediately tested afterwards. It's working. So we have prevented a lot of relapse. We prevented a lot of deaths for sure because people can test them now, do the ridiculous price every day if they want to, and getting the results in days and weeks and can adjust their treatments. So yeah, AI is here to stay for sure.
Absolutely. I'm glad you came up and say that, Danielle. Well, I mean, my company, you can check it out online, yatiribio.com, YATIRI bio. We use proteomics
Ah,
to figure out changes in cancer cells before and after uh, administration of the chemotherapy, and based on responsiveness on samples, we can predict if you have certain biomarkers, you're going to be responsive or non-responsive.
yeah.
We have calculated based on running through clinical trial data of drugs that failed, that by doing this, success rate goes up by a factor of 3
Mhm.
because you're going to be running the clinical trial only in responder populations. And so with the same amount of money, a pharma company can run 3 trials with a much better outcome. And we have done small proof of concept trials. They've been golden, and now in a couple of months, we're going to go into a very large investigator-initiated uh, uh, trial at a large cancer center to where the patient population is selected using our methodology. We already have results of clinical trials of the same compound without our things. So everybody's really excited that we're going to get much better results. So yes, AI works.
And
AI
it
is
makes
an
sense.
amazing tool, but you have to know how to use it. You have to know what questions you're asking it and what results you're getting out of it and what do those results mean.
Yeah, Sam, you choose a very difficult field because with proteomics you have so many secondary and tertiary effects. You really don't know the causal one,
Yeah,
and that's the super difficult. So we are the group. Then we actually try to find the markers. We're doing RNA, DNA, but also epigenetics and proteomics. We didn't touch yet because we want to like to see the first responses. We want to find the early onset targets,
yeah,
and not actually then the results. So we're more in the diagnostic part right now, but we currently with another company we're working on drug discovery or drug responses, and there we are going and actually making hundred thousands of experimental tests
okay.
using organoids. So actually, would be interesting to work with you guys
Yeah,
because
yeah. Check out our website. I mean, the website right now is not very informative because we haven't updated it in a while. Uh, we've been busy with other aspects of this,
same here.
really
Web
getting
page
ready for
don't
this
say
clinical trial.
anything.
And
Yeah.
we're also starting our Series A, so everybody's been really busy with other things. But you kind of will get a kind of a get feeling for what it is we're doing now, and we're always looking for collaboration and you know
Yeah, I think
all of that.
that'll be interesting. So our webpage, the same thing. We're too busy for the webpage, and nobody looks at the website, especially if you're a scientist. Nobody looks at the
Yeah,
webpage.
no,
This is
it's
too old information. Yeah, you can always reach me out. You can always Google me. You find enough information online about my name. I going around here with my real name. I don't see why I should use some
yeah
fake
that
ID.
I am.
So you can find my LinkedIn and then you can reach me out there. It's the
I
easiest
have my
way.
bar license number on my
Really?
bio. So yeah, I mean both at the patent office and California bar. So there's not much I can do to hide. I have
Yeah.
doxed myself. So
So
what it is.
also I like to have a dual career, so I'm like more than one, but I'm not only an entrepreneur, but I'm still affiliated in the University of Tübingen. We're the second largest genome center in Europe. So we have tons of patient and tons of data, and we have literally every machine, but you can imagine our genome center, like literally everyone. Although even those ones which are going to be released, all the prototypes in the next 4 to 5 years. What I want to ask you, I mean, proteomics. I'm friend with Nano and they're not existing anymore. Bruker now, they're Brukers.
Uh,
But spatial temporal sequencing, right? And
wait,
proteomics.
Varian, is that who we were
No, the
talking about?
nanostring was it before? Now that got sold
Oh,
to
to
Bruker.
Brooker. Oh, okay.
Yes. And the chief scientific officer, a good friend, and I've been working with them some projects. And I mean, like we worked on GeoMax That is special sequencing. So that means single cell sequencing. So you cut open the tumor, and then you can sequence every cell in the tumor, DNA, RNA, and proteomics for every cell. And you can distinguish between the membranes, distinguish the nucleus between like little parts. And then if you're looking at tumor tissue, you can the outer tissue, you can inner tissue, you can looking for heterogeneity in tumors. And I thought that would be very interesting for you. And a machine called GeoMax, but there are many others. But these spatial temporal sequencing, temporal for, before, during, and after the treatment, spatial like 2D over the tumor cut, that would be very interesting for you because then you not only can see where the treatment is, but where also the issue is. For example, transport, etc. I would look in that. And then you need definitely AI because the data is overwhelming.
Very good.
But
So
Welcome, Pharmas.
thank you, sir. Thank you. So I'm asking a question about the AI in medical, medical fields. So I'm a radiology technologist. So how can we make AI? We can use AI in our field.
Well, I think AI in radiology is already being used in terms of identifying anomalies in the scan. It can identify anomalies that the radiologist's eye may or may not be able to see immediately. But from what I understand,
and in
and
diagnostic, as well, because we're already
yeah,
live in hospitals.
that's the area that I've heard where the issues are in the diagnostic area that uh, still humans are better at diagnostic
Oh,
than
not
AI
anymore.
is.
Not anymore. We have no Edmore. That's what I said before. Like we are had certification process for 4 years, 12 years in development, and we spin out, and we're live in hospitals. So the government and government funded hospitals
Yeah.
with 80,000 patients, reimbursable by health insurance companies, and fully certified.
Are you
And
in
we're
the
better.
US or?
In the US is a competitor for us, but we're currently expanding to the US. But currently we're in Europe. And we're also working with the German government
Yeah.
because we have so much data. We developing, we helping with the guidelines for AI security, patient security, which is very important. And we're fully compliant with patient security.
Yeah.
So yes.
So,
Mhm.
So
so for example, other guys told me maybe our department, our field will not be able. So will not be able working with AI because it's about the diagnosing, and so because the doctors and the other medical staff are in the hospital will not be able to use AI. But the other field like computer science and even I don't know other fields will be in touch of AI. But in medical field, it's I think it's so difficult to use AI tools. Am I right?
no, that is
No.
absolutely, I think, not true. In fact, radiology was one of the first disciplines that people thought could be, in terms of diagnostics, not in terms of doing the procedure, but in terms of diagnostics, it could be all AI. And so, I mean, Daniela and I maybe kind of like about 6 months apart from each other, or our sources are
Yes.
in terms of diagnostic ability. So you're much more in the front lines of that than I am. So my information is old. So
But you
let's say,
also You're in
Uh,
proteomics. You're very far in this. I have no
No, no,
idea. And you're but you're
I
doing in
was talking to
proteomics. Amazing.
radiologists about
Uh,
this,
So,
but this was about 6 months or so ago that they were
so
saying this.
yeah, maybe in European countries, United States will be using AI more. But so I'm living now. Currently, I'm living in Afghanistan, and this situations by the government, we are not able to use AI technologies for ladies and new generation in Afghanistan. It's really difficult. The situation will be so badly running right now in the
Yeah,
world.
that's a that is a political question, not a technological question, because even if you don't have the facilities there, you can email the the results to a radiologist somewhere else or an AI machine somewhere else, and they can do it and send it back to you. But whether you have access to kind of global markets for technology, that becomes a political question, which we can't really answer that.
And also, you have to think about there are several ways. So we're not at the level we talk. We upload patient data in ChatGPT. We would never do that.
No.
We have our own AI and our own servers, which
Yeah.
is at their country. So we also work a lot with on-site AI. So like basically a server on-site, which have 100% patient security. So there's absolutely no reason to actually not work with AI. It's purely if they still say no to these on-site premises solutions where highest patient security, and trust me, Germany is one of the hardest countries in patient security. In US, it's so much easier. And by the way, we're working currently with a contract with 50 hospitals in the US, and it's so much easier than Germany,
Yeah.
for example. But I think it's a pure political reason because technology and patient security and all these steps for secure measurements, AI, that's done in really high professional healthcare companies and universities, not in general, of course.
So let's go back to generic drugs. Do we have any? It seems to be that nowadays it doesn't matter. You can be talking about, I don't know, the Sasanian Empire, and people will talk about, well, how did AI affect the
Well,
fifth
the thing
century
is,
kings?
I think belongs
AI
to
is.
the Iran.
What was that? Or
I was saying AI is going to be important for generic drugs because they're going to utilize those methods to make them faster, like to the market. Like you were saying, yes, you were saying earlier how they like wait until the day the patent expires and then they're ready to ship out with all this distribution network. They're ready to go right away. You know,
Why would that speed up? Isn't that a delayed starting line that by law? How could that be sped up?
so that AI can help with drug development, there's no question. But the question is, at what level? What can it do? And the way I see it being used is that it is not giving us uh, things that we didn't know before. But it is doing things that it would have taken us a long time to do. So it basically is speeding up all the thought processes that we had before, right? We knew X, Y, and Z need to be done. It would take 5 years to do it. Now with AI, we can do it in 10 days. But it hasn't changed how we think about things
There
in
is
drug
one
development.
There
Yeah.
is one distinguish, and I'm pretty sure nobody knows about that, but please Google me, you find tons of information. So we use AI, for example, a new thing which a human like with our brain would never could accomplish. Also, we never thought in that direction. I work in space biotech. What it means. So every one of you know astronauts. Astronauts are very healthy. And when they go into space, they suffer on these harsh conditions of microgravity, which cause microfluid shifts, radiation cause cancer, etc. Right? So what we do is we sequence and test and analyze astronauts before the flight, during the flight, and after the flight. And what we can see early onset markers of, for example, diseases like Alzheimer, right, or cancer or heart cardiovascular disease. But then in the next, we actually use AI to find targets, mostly already existing drugs and market or repurposing max. And we found drugs which before was repurposed for psoriasis, like a skin condition, is one of the, was killing 80, 70 to 90% of the cells of triple negative breast cancer, one of the breast cancer where don't have treatments yet. It is so complex that a human brain would never have found it in 10 years. But we found it in 2 months. It was a very, very difficult AI program. So we now using like very resources, like there are not many resources we need. We
So
just
wait,
Mhm.
how was what you did a paradigm shift? It wasn't a paradigm shift. It just did something that you knew needed to get done.
I
You
was
did
not
it a lot
finished.
faster.
I didn't finished. The second one, what we saw is protein structure. Sam, you know about these things, right? We AlphaFold and all these. There are already so many other programs. They can actually predict, let's say, from a part of protein, four different kind of structures. And a protein, if you, for example, have a loop, that would be a perfect. There's a loop, and it's predicted for being a perfect target, right? But we can't. We think we found all protein configurations. We know the amino acid sequence. We know pH. We know temperature, etc. So AlphaFold and but is predicting 11 conformation changes of this protein. That means much more access point for new targets. And we proved them like in experiments, and they were true. But by our knowledge today, only four would be possible. The 11 seems impossible, but at the end, they were true. And that was mind blowing for me how much potential we still have, especially in proteomics, Sam, what you're working, that conformation changes the same amino acids, same like metal content, the same salt concentration, and temperature is also important, as you know, charges. It was mind blowing, and it taught us a lesson that we don't know anything about protein conformations yet, because it was true. It worked, and that is something with completely new, which also leads to then we could find new drugs that we could even prove that these loops which are predicted were accessible for drug treatment, for drug responses. And I think, Sam, if you're your company, you could actually look for these things. I don't have the bandwidth to do it, but I would be curious what you think about that.
Well, I mean, I'm not going to get into like uh, kind of scientific experimental design, but I was going to say that still what you discussed was not a paradigm shift. So, for example, if I say uh, there is a lot of water over here, I need to dig a trench to get the water out, and I have a spoon, and then you come out with a backhoe and you do it much better, much faster. You can actually dig out places I couldn't have dug out. That is not a paradigm shift because it's still we're digging a ditch to get the water from here to there. And so the thing is that you are identifying secondary structures within the tertiary structure using AI. Well, if you were to come up with a new secondary structure that we didn't know about, something that,
Yes,
let's
that I'm
say
talking about. That was I'm
it's
talking
a
about.
it's like
New.
a gamma screw, right? We don't, but what you're saying is that you're just identifying where alpha helices and beta turns are within this. So that is something that we could have done using structure elucidation techniques that we had before, but it would have taken us a long time to do it. But have you found uh, motifs that we didn't know about before,
Yeah,
right?
I found motives in another experiment, which it's a
So
totally
what are
different.
they?
Okay, so do you know the NASA twin study? Do you ever heard of this?
The what?
The NASA twin study. We
No,
in 2000,
I
I
don't.
worked there for 3 years together. I was research director in volcano medicine in New York for a couple of years, and we worked with the twins. So they're identical twins, Mark and Scott Kelly. And Scott Kelly went to space for 1 year, and we all know space is an escalator for diseases. And his identical twin brother, Mark, stays on Earth. So perfect, perfect things. And we had like 5 of the 12 founding team of the NASA twin study. We were looking into aging, right? So how you can predict age in a human being, it's telomeres, telomere length, one of them, and methylation patterns, of course. And in telomeres, if you age, every one of you, you have telomeres at the end of the chromosome, they're getting shorter and shorter, like, like a salami or somebody put pieces off. Yeah, you're getting shorter and shorter and shorter with every year. And in space, they suddenly got longer. So by definition, astronauts in space getting younger, much younger, which can't be because the harsh conditions actually should aging faster. So that time we published 3 publications. You can file them. They're all in the Cell. They're all Cell papers, Nature and Science paper. You can just Google my name, PubMed. You find those. But then one thing what I was completely blown away is that we have like this ultra long read sequencer, like nanopore sequencing. You can do whale reads, called it, like several 500 kb long stretches. And we decided to make a really long read to sequence into these telomeres in. These telomeres are very difficult to sequence because and we found motifs there. It's not repetitive elements anymore. They're completely new motifs. And without AI, we wouldn't have been able to identify them. And that was something like, because it was just too long and too much data, but that was mind blowing because we have no idea why the heck telomeres are supposed to have binding sites and why they're longer in microgravity at all. And they shrunk again. By the way, now they only were long during the flight, and when I came back to Earth, they're getting small again. So, in definition, like if you say telomere is really age, the astronaut was old, getting space, getting young, and getting back old. And there was something like, which until today, although we have like 3 publications at least in high impact journals, still have no idea. I literally have no idea what that means. But there was something completely new. And I wish I could work. I still work in astronaut health,
Are,
but yeah,
Daniela, are there other things that you could correlate with age other than telomere length? What if
yes,
it's just something under no gravity that telomeres expand, or are there
David.
other hypotheses?
Yeah, David, you have an excellent question. So the other one is, which we know that has nothing to do with age, is because we did also the metallome. Methylation, you can predict the age of a human being by the methylation pattern. The methylation shows that the astronaut stays the same age, actually getting a little bit older because of all the stress. So one of the thing is where you can see longer telomeres is cancer, but also, but I have no recall of one case. But also think about, David, and it's a good question, is population. right? Cells are population. What's about if all the cells with shorter telomeres just died and a population of longer telomeres resist? But still, the telomeres were so long. We never saw this on Earth. There was another thing we never ever saw in all the human history. We saw a massive death of mitochondria. So I found this is first author paper in Cell. You can check it out. We found super high amounts of mitochondrial RNA, DNA in the blood plasma. Never saw on this level before, and that will explain a lot of the aging effects general in astronauts, like why the early onset on neurogenetic disease, for example.
Has there been any space research on organ transplant rejection, that phenomenon, and anything around that?
Oh, that is so. No, well, the thing, oh my God, David, you're asking such excellent questions. Well, we think there's a lot of very similar to immunoresponse. So the immune system is completely really down in space. So that's why they're getting so many infections. But that all comes down to, and then maybe you're an expert in organ response. So it's all comes down to hypoxia, so the lack of oxygen. So, well, how that can be possible? The ISS space station provides 18 to 21% oxygen, right? But still, astronauts suffer, and you can see clearly in their data, in their RNA proteomics. By the way, Sam as well. We always do proteomics a lot there, and the effects of lack of oxygen, and that's simple a physical constraint, convection. So on Earth, when we breathe oxygen in, we get it from the air, sure, but if we breathe out carbon dioxide, it's more heavy than air. It falls down to our feet. But in space, we don't have gravity, so the carbon dioxide sticks to the face. And that's why if astronauts are not in a well-ventilated area, they suffer on oxygen. They're basically not getting enough oxygen. That lead to hypoxia, that lead to mitochondrial diseases, and that is also responsible why their immune system is so down. And that these insights, what they're currently testing, and there are currently a lot of experiments with cell cultures up there, is hopefully is an answer to how we can prevent these issues we just described with organ repel, effects of organ transplants to reduce them.
The rejection is the
Rejection, yes, exactly. And I think like the fact is that we can study these things in space within months instead of years on Earth is mind blowing. So we use this a lot. But you can just Google my name and PubMed. You'll find a couple of publications also with immunoresponses.
All right, so we kind of moved away from the main topic a lot. I think because of the two part of this. Amash, what do you think? We should stop this room and open up an after party and continue the conversation
Idea so
there.
that the recording will be available to people who are interested in the topic. Sure.
Yeah, yeah. So thank you all for joining us, both who joined us here on Clubhouse and at YouTube. Thank you for being here. And don't forget, every Friday at 9:30 Pacific, 12:30 Eastern, we're going to have our scientific discourse room. Come back and check us out. Better yet, become a member of the house so